This video is an introduction to enzyme pH ranges. Enzymes function in a narrow pH range. Above or below that, they become destructive. Enzymes move electrons and protons to different locations within a molecule. These movements cause the bonds to break between atoms, which change the structure of the molecules. This is similar to the magnetoelectric bonds in the magnets seen in this video. Because the movements of electrons and protons is so important to enzyme activity, enzymes are very sensitive to pH. The conformation of an enzyme is maintained by interactions between the various amino acids that compose it, and this conformation is sensitive to changes in the enzyme's environment. Each enzyme is designed for a specific purpose. Each enzyme works within a small pH range. There is a pH at which its activity is the greatest, known as the optimal pH. This pH maintains the shape of the enzyme and therefore its effectiveness. Two important influences for enzymes are pH and temperature. When an enzyme's conformation is altered because of pH or temperature variation, the enzyme may no longer catalyze the specific reaction it was designed for. A change in pH can make and break intra- and intermolecular bonds, changing the shape of the enzyme and therefore its effectiveness. An enzyme is said to be denatured when it loses its functional shape. A special region on the enzyme called the active site has a shape that fits with the specific substrate molecules. These interactions between the substrate and the enzyme strengthens or weakens the magnetoelectric bonds in the substrates. These magnetoelectric forces interact with the substrates, leading to the formation of a different molecule. As a result of the chemical interaction within the active site, products are formed or broken apart. Enzymes are beneficial within their specific pH ranges, but turn destructive in a terrain that is more acidic or more alkaline than their specific ranges. Changes in pH may not only affect the shapes of the enzyme, but it may also change the shape or charge properties of the substrate so that either the substrate cannot bind to the active site or it cannot undergo changes. Acidic shifts cause desiccation or denaturation of tissue proteins, causing shrinkage. Alkaline shifts cause a process that involves saponification of fats and solubilization of proteins, causing enlargement.